Well, I think my position is really starting from a point of clinical need, and that should be really what drives what we do in sort of genome screening. So obviously there's the sort of genome-wide association type studies where you're just looking at SNPs across the genome, and there's not really that much utility at the moment in using that kind of screening. But ultimately when we do whole sequencing, there's going to be a lot of usefulness, particularly looking at rare variants and understanding inherited diseases. There are enormous ethical implications. I think one of the major issues at the moment is trying to understand what we do with incidental findings, so things which we find which we weren't necessarily looking for, we're not even necessarily sure how to understand them, and people don't necessarily want to know them either. So trying to understand what sorts of things do we feed back to patients or research participants that wasn't in the initial intention for testing. There's obviously other issues relating to family members and the implications for family members versus the individual, and also for informed consent and really ensuring that patients and individuals understand what it is that might happen as a result of testing. So the implications for patients are fairly broad. In terms of genome-wide association studies, the main implications I think are going to be an understanding biology and the understanding of how disease mechanisms work. So it's going to be developing new treatments and new targeted drugs and so forth. In terms of whole genome sequencing, we don't really know yet. I think there are major implications for understanding inherited diseases and rare variants, but also in cancer genome sequencing, and really again stratifying treatments to suit the patient. Genome-wide association studies, I think there's potential for those being used to stratify entire populations so that you could categorise people into high or low risk categories, and then you could target preventative interventions such as screening at the high risk group. So that's a possibility there. In terms of the whole genome sequencing, there's obviously single gene disorders, so something like cardiovascular diseases. There's a number of inherited cardiovascular diseases across different genes, and we're not necessarily sure which one to test in each time. So if you could do a whole sequence, you'd be able to look for all of them at the same time, and that could potentially save a lot of lives. There are a number of issues, I guess, that need to be worked through, particularly this issue about incidental findings. There's obviously big concerns about genetic privacy as well, and who has access to the data. And really, the major thing for whole genome type sequencing is going to be data. It's an absolute deluge. We're talking about 3 billion base pair per person. How do we store that data? Who has access to it? How do we actually interrogate it in an evidence-based way so that we try and understand what it is that we're actually going to feed back to the patient? I think those are the really major challenges, and if we can't overcome those things first, then it could really delay the translation of this new science into the NHS.